Okay, our cell signaling animation project will be a demonstration of a paracrine regulator. First, we start with two ligands in the secreting cell. These ligands act as the molecules that send signals to another molecule. The ligands will come out of the secreting cell into a neighboring cell, which we call the target cell. And they will connect to these two tyrosine kinases. Now, this causes them to clunch together into what we call a dimer. And in that process, 6 ATPs, these 6 ATPs will come and attach 6 phosphates. There's only 3 right here, but in reality, there's 6 phosphates that connect to each section of the tyrosine kinase. And then after this happens, we have these inactivated relay proteins right here. Once the tyrosine kinase has been activated by the signaling molecules, they will then activate these relay proteins and turn them from inactivated to activated relay proteins. And so we left off with these two relay proteins getting into an activated form. Now, once these relay proteins are activated, when they recognize the tyrosine kinases, the transduction process now starts. And then what these two relay proteins do is they activate the enzyme adenylyl cyclase. And adenylyl cyclase, after that, it uses an ATP.
Oh, that's right.
And from the ATP, it breaks out 2 phosphates. And those 2 phosphates are used with ATP to create cyclic AMP, which is shown right here. Cyclic AMP then activates these 2 kinases. It first activates kinase 1. And what kinases do is they add phosphates to inactive kinases, thus making them active. So kinase 1 will activate kinase 2, giving us phosphate to kinase 2, which turns kinase 2 into an activated kinase. And this process keeps repeating. There's only 2 shown here, but in reality, there could be 5, 7, maybe 10 steps in what's called a phosphorylation cascade, where kinases keep bringing phosphates from each, giving phosphates to inactive kinases, rendering them active. And where amplification comes in is that every time a step goes from, let's say, adenylyl cyclase to cyclic AMP or cyclic AMP to another kinase, it amplifies the process. One adenylyl can create sometimes 10 cyclic AMPs, and each step can create 10 or maybe even 100 copies of the next step, which is why one molecule of, like, for example, epinephrine can make 1, 10, 100 million glucose molecules. And then it's the opposite of kinases is the phosphatases. What phosphatases do is they remove phosphates from other things, which kind of reverse the process, which allows this whole thing to start over again. And yeah, I also mentioned that second messengers are non-enzymes that kind of act as like a signaling molecule. In this case, the cyclic AMP is a second messenger because it starts the process of this kinases, which starts phosphorylation cascade. And how can they be regulated, you say? Every step in this process has certain inhibitors that can be turned on or off at certain times. When the inhibitors are on, the whole process stops. And at each one of these processes in the steps can be regulated by some inhibitor or the other. So whenever this process needs to stop, it can be regulated by the use of inhibitors in any step that's necessary. And that's basically the overview of the entire transduction process. So after all the steps of transduction, the very last protein kinase gets activated, and it triggers cytoskeleton changes in this case. These are 2 fibers in the cytoskeleton, and an example of how the cytoskeleton can change because of these reactions is, let's say, these 2 fibers can get connected as a result of all this transduction in this process.